The third recipient of this year's Paul Marks Prize is Doujia Pan, and to introduce his body of work and him is Catherine Anderson. Catherine.
I'm really privileged to introduce Doujia Pan today. Doujia is a professor of molecular biology and genetics at the medical school at Johns Hopkins. He's also a Howard Hughes investigator. Doujia's career has been a wonderful story of basic biology moving through from fruit flies to mice to humans. So Doujia started his scientific career as a graduate student at UCLA, also my alma mater, where he worked on a gene I love very much called dorsal, and he did biochemical studies to show that this dorsal NF-kappa B protein could either activate or repress transcription depending on its context. He then went to postdoc with Jerry Rubin in a very exciting time in Jerry's lab where they were using powerful new tools to identify more of the genes that control intercellular signaling. So they were using mosaic analysis to make small mutant clones, and use that as a way of identifying new genes that were important in controlling signaling in the fly eye. In that context, he became involved in a characterization of a gene called Kuzbanian, a characteristically fly designation for a gene now called ADAM10. Doujia showed that Kuzbanian was required for proteolytic processing of Notch. So this is the very first example where it was shown that Notch needed to be proteolytically processed to become active. And so from his mutational and biochemical analysis, he showed that this protease did one of what we now know are 3 different cleavages in Notch that are required for activation of Notch. He then established his own lab at UT Southwestern, where he continued to do mosaic genetic screens in the fly to identify more components required for intercellular communication. And at this point, he decided to focus on growth, which is something that had been understudied. People had studied a lot about cell type specification in flies, but growth had really been rather overlooked. And so he started looking for mutations that affected ability of cells in clones in the eye to grow. And he found himself studying molecules that are familiar to this audience, like P10, TSC1, TSC2. And he used the power of Drosophila genetics to put these together into a functional pathway in vivo to understand how these molecules work together to control growth in the fly. And that pathway turns out to be rather more complicated. It's not just a simple linear pathway, but the genetics was powerful enough to tease that apart. And I use these papers when I teach in the GSK core course for cancer biology. Then about 10 years ago or so, he moved to Johns Hopkins, where he continued to work on growth control in Drosophila. And it was about that time he identified yet another growth control pathway that had not been previously discovered, and that's the Hippo pathway. So in parallel with some other fly labs, he is the person who really discovered this new growth control pathway and put together the players and came up with a pathway that controls growth that was a previously undescribed pathway that was only discovered through this application of Drosophila genetics. And that's what he's going to talk about today. And so we're excited to hear how he's pushing this pathway from flies to mice to human biology.
Thank you, Catherine, for that very generous introduction. It's a real honor to be a recipient of the Paul Marks Prize, which speaks to the legacy of Dr. Paul Marks, both as a scientist and as a leader of biomedical research. For myself, it is a real privilege to follow the footsteps of many of the previous awardees of this prize, which include my longtime friend and scientific role model, Sheldon Wang, who happened to be in this audience, and he gave me a big surprise by showing up like 2 hours ago. I should say that this prize not only recognized the work of my laboratory, it is also an important recognition of an emerging field of study and the contributions that are made by many labs in the world to this new signaling pathway called Hippo. And so I feel very privileged and to receive this prize, and I'm very grateful for the selection committee. I'm grateful for the Sloan Kettering Institute and for giving me this tremendous honor. And last but not least, I'd like to thank my family, my scientific mentors throughout my training, and my colleagues and many collaborators, which enabled us to study this new signaling pathways, both in flies and mammals, and that really allowed us to translate our insights from Drosophila to cancer biology. With that, what I'd like to do today is to give you some of the highlights of our work in the past 10 years about the genetics of growth control. So I was trained as a developmental biologist. As a developmental biologist, we are interested in the fundamental problem of how cells acquire their cell fate, and also how organs are built, both in terms of their fate determination as well as their size control. If you look in nature, you know that the difference in organ size, organismal size, is one of the most visible features that distinguish different species, or even among individuals of the same species. So as such, it has been a long-standing interest by developmental biologists to understand how the size of organ or organism is determined. So the key question we ask are the following. First, how is the size of an organ determined in development, homeostasis, or regeneration? Secondly, since a hallmark of cancer is unregulated growth, we are also interested in understanding how does dysregulation of the size control mechanism contribute to tumorigenesis in mammals. So my lab took the following approach. First, we used Drosophila, the fruit fly Drosophila, as a genetic model to uncover size control genes. We then use a combination of fly genetics and biochemistry to study the function of these genes in the context of cell signaling networks. We then apply the insights we learned from the Drosophila to investigate whether the size control mechanism we uncovered in flies regulate tissue homeostasis in mammal, and how the dysregulation of such mechanisms contribute to tumorigenesis. It's through this concerted efforts of using both Drosophila and mammals that led to the discovery and the characterization of this new pathway I'm going to talk about today, which is the Hippo signaling pathway. I will tell you that the Hippo pathway is a central regulator of tissue growth in animals ranging from Drosophila to mammals. Actually, the first 4 components of the Hippo pathway were identified as tumor suppressors. And these 4 proteins include 2 kinases, WRTs and Hippo, and 2 adapter proteins, Salvador and MATS. These tumor suppressor proteins were identified in a 10-year span by a number of labs, including our own, based on a very similar cell autonomous overgrowth phenotype in mosaic flies that contain homozygous mutant clones in an otherwise heterozygous genetic background. But it was not clear, immediately evident from these genetic studies whether these 4 proteins define a single biochemical pathway. Through a series of biochemical studies, my lab demonstrated that these 4 tumor suppressor proteins actually function in a single biochemical pathway that ultimately impact transcriptional regulation of growth control genes. We show that these 4 proteins form a kinase cascade in which Hippo, together with Salvador, phosphorylate and activate its downstream kinase, WRTs. WRTs in turn phosphorylate and inactivate a very important oncogene called YORKI, which is identified by my lab. YORKI really represents the ultimate target of this phosphorylation cascade because gain function of YORKI, either in mutant clones or overexpression throughout the organ, can completely phenocopy any of the loss of function mutations for the upstream tumor suppressor genes. And furthermore, we demonstrate that the YORKI actually functions as an oncogene, as a transcriptional co-activator by binding to a DNA binding transcription factor called scalloped or SD. And this transcription factor complex can normally drive the expression of pro-growth genes, such as anti-apoptotic genes, DIP1, and cell cycle regulator cyclin E. But normally, the activity of the Yorkie-SD complex is under check through Wurtz-mediated phosphorylation, because phosphorylation of Yorkie retains Yorkie in the cytoplasm away from the nucleus, where it can drive the expression of pro-growth genes. So therefore, when you lose any of the upstream tumor suppressors, Yorkie is unphosphorylated, is constitutively in the nucleus, and resulting in increased transcription of target genes, such as DIP1 cyclin E, therefore resulting in dramatic tissue overgrowth, as I show here. Building on insights from Drosophila, we then demonstrate that the mammalian counterparts for the Drosophila Hippo pathway components actually form a similar kinase cascade in the mammalian cells, which converge on gene transcription and organ size control. For example, we demonstrate that using a transgenic mouse model that's regulated by tetracycline, and specifically in the mouse liver, we show that overexpression of YAP, which is the mammalian counterpart of Yorkie, in the adult liver can result in a dramatic 5-fold increase in liver size within 4 weeks of YAP induction. These mice went on to form focal hepatocellular carcinoma after extended period of YAP induction. So if we induce YAP induction right at the birth, these transgenic mice then form a widespread hepatocellular carcinoma throughout the whole liver within 8 weeks of age. So these studies demonstrate that the Hippo pathway play a conserved role in mammalian organ size control and tumor genesis. Based on the potent oncogenic activity in the YAP transgenic liver, we ask whether the YAP protein is also upregulated in human cancers. Using a tissue microarray, we examine a collection of primary tumors from lung, ovarian, pancreatic, colon, liver, and prostate cancer. We found that the majority of these primary tumors show overexpression of the YAP oncoprotein. These results are consistent with independent studies from cancer geneticists, which include Scott Lowe and Michael Overholzer, who are now here at the Sloan Kettering, which their study demonstrates that the YAP gene locus is amplified in a wide spectrum of mouse and human tumors. In fact, the overexpression or nuclear accumulation of YAP has been used as an independent predictor of prognosis for several human cancer types, such as liver cancer, ovarian cancer, and colorectal cancer. In our efforts to try to understand the transcriptional regulation of the Hippo pathway at the very end of the Hippo pathway, we recently discovered a co-repressor that directly binds to the DNA binding transcript factor at the end of the pathway, either SD in fly or the TAD protein in the mammal. So we discovered a co-repressor called the TGI that binds to SD, and its counterpart in mammal, which is called the VGO4, which can bind to TAD. It turns out that the SD protein actually lives a double life in transcriptional regulation. It can either activate transcription in complex with Yorkie or repress the same target gene in complex with its co-repressor TGI. In fact, the default state of SD is transcriptional repression, and the Yorkie normally drives tissue growth by antagonizing this repressor function through a relief of repression manner. The discovery of this extra layer of transcriptional regulation is important because we can take advantage of this phenomenon to try to intervene the oncogenic activity of YAP. For example, in the mouse liver, the overexpression, I already showed you, the overexpression of YAP results in this massive development of liver cancer. However, this phenotype can be completely reversed by overexpression of the TAD co-repressor VGL4. Another way to intervene the oncogenic activity of YAP is to develop small molecule inhibitors against this oncoprotein. Since I already told you that the SD or its mammalian counterpart TAD are obligatory DNA binding partners for Yorkie or YAP, one way to interfere with this oncogene is to try to identify small molecules which can disrupt the physical interaction between Yorkie SD or YAP TAD. Recently, by screening through FDA-approved drug, we identified a small compound called the verteporfin, which can physically inhibit the interaction between Yorkie and SD or YAP and TED in vitro. But most importantly, in vivo, we demonstrate that the administration of the verteporfin can significantly reduce YAP-induced liver overgrowth, demonstrating that the YAP, this inhibitor, has in vivo efficacy. So far, I've told you about the Hippo signaling pathway from Hippo all the way down to the transcriptional regulation. And in the last few years, the major efforts of the field has been trying to understand how the Hippo signaling pathway is regulated by upstream signals. The general idea here is that the Hippo pathway is somehow activated towards the end of organogenesis in order to stop organ growth at the appropriate size. But the major question in the field is to try to understand the molecular nature of the signal which turn on the Hippo signaling pathway at the appropriate time. The very first insight into this question was actually provided by George Holder's lab back in 2006. George Holder's lab identified two firm domain containing proteins, expanded and Merlin. And these proteins are associated with apical junctions as well as cytoskeleton. I should mention that the FVRM domain containing proteins are typically found in proteins which function as the membrane cytoskeleton linkers by interacting with both plasma membrane and underlying cortical cytoskeleton. However, the specific mechanism by which expanded and Merlin impact the Hippo kinase cascade was unknown. It was also unclear how expanded and Merlin are targeted to apical junctions. Also, through an unbiased genetic screen, we recently identified two additional upstream regulators of the Hippo pathway, which function in conjunction with expanded and Merlin to activate the Hippo pathway. These two proteins are called CRAMPS and KIBRA. So CRAMPS is a homotypical cell adhesion molecule which contains a very large extracellular domain containing 28 EGF repeat, as well as a very small intracellular domain. We found that the intracellular domain of CRAMPS contains a canonical firm domain binding motif. And in fact, we demonstrate that this firm binding motif can directly bind to the expanded protein. And this interaction is required for the localization of expanded to the apical junctions. The KIBRA protein is a C2 and WW domain containing cytoplasmic protein. It's also localized to the apical junctions. We found that KIBRA physically interact with expanded and Merlin and function in a combinatorial manner with expanded and Merlin to activate the downstream Hippo signaling. For example, if you look at single mutations of KIBRA or Merlin, they actually result in a very mild overgrowth phenotype, which is not very visible at this kind of resolution. But importantly, when you make a double mutant combination between KIBRA and Merlin, now you can see a very severe overgrowth phenotype, which can be shown by the external morphology of the adult eye, or the presence of these extra layers of cells which separate neighboring ommatidia. And normally, there's only a single layer of cells separating neighboring ommatidia. Now you can see multiple layers of cells that separates neighboring ommatidia. So this kind of a phenotype actually resembles the inactivation of the kinase cascade at the bottom half of the pathway, suggesting that these proteins function in an additive or synergistic manner to activate downstream signaling. So one of our major efforts in the last few years has been to try to understand how these apical junction-associated proteins regulate the Hippo pathway at the molecular level. We are particularly interested in understanding one of the proteins in this complex, which is called Merlin. Because the Merlin is so far the only Hippo pathway component whose human homolog has been linked to a hereditary cancer syndrome. Merlin was actually identified exactly 20 years ago as a tumor suppressor gene underlying this human disease called Neurofibromatosis type 2, or NF2. That is why this protein can be also called as named as NF2. The NF2 disease is an autosomal dominant disorder that affects 1 in 60,000 people. The disease manifestation includes the development of schwannomas, meningiomas, ependymomas, and cataracts. And the major treatment for this disease consists of surgical removal of the tumors or the cataract. There are currently no molecular target therapy for this disease. Over the last 20 years, many studies, including those from Filippo Giancoti's lab here at the Sloan Kettering, has linked the Nf2 or Merlin protein to cellular processes which are relevant to tumor suppression, such as contact inhibition in culture cells. However, it was not clear which effector pathways are controlled by the Nf2 or Merlin protein. We recently identified a molecular mechanism that directly link the Nf2 protein to the Hippo signaling pathway, which will be summarized in the next slide. So we found that actually the Nf2 or Merlin protein can exist in two conformational states, either a closed inactive state or an open active state. The open or active state of Nf2 can directly physically interact with WRTs or the mammalian counterpart called LATs, and directly recruit WRTs to the plasma membrane. Once WRT is on the plasma membrane, it can then be phosphorylated and activated by the upstream Hippo sourdough protein kinase complex. And once WRT is activated, it can then phosphorylate the downstream target, Yorkie, and to keep Yorkie outside the nucleus. That's how WRTs can inactivate Yorkie. And I just show you a tiny bit of data to demonstrate the relevance of this interaction. So we found that the physical interaction between Nf2 and the WRTs is actually mediated by the firm domain of Merlin and a very short N-terminal conserved motif in the WRTs LATs protein. And interestingly, most of the disease-associated mutation for the Nf2 disease are found in the firm domain. We therefore took a collection of human disease-associated mutation for the Nf2 protein and tested its physical interaction with LATs protein. As you can see here, while the Y-type protein combined robustly to the LATs protein, none of these human disease-associated protein can physically interact with LATs. Conversely, we demonstrated if you mutate this very short sequence motif in WRTs protein that's required for its physical interaction with Merlin, now this WRTs protein can no longer function as a tumor suppressor in vivo. One of the interesting observations that came out from this study was that using cell culture, we found that actually the transition, the switch between the closed inactive to the open active conformation of Merlin is actually regulated by the actinium cytoskeleton. So for example, when we treat the cells with the inhibitor of the Rho small GTPase or inhibitor of actin polymerization, we find that we can activate the Merlin protein and promote the physical interaction between Merlin and its downstream target WRTs. So one of the direct implications of this finding is that the Merlin protein may actually function as a molecular sensor that transduces mechanical signals to growth control. Of course, there's still a lot of study we need to conduct in vivo to fully substantiate this model. Another implication from this work is that the YAP protein, which sits at the very end of the Hippo pathway, may represent a critical target for the intervention of tumor growth induced by the loss of function of Nf2. Indeed, we found that the Nf2 deficient phenotype in many mouse tissues are exquisitely sensitive to the gene dosage of YAP. In the mouse liver, for example, loss of function of Nf2 leads to liver overgrowth and liver cancer development, which mimic the YAP overexpression phenotype I showed you earlier. Strikingly, this phenotype can be almost completely suppressed by even a heterozygous mutation in the YAP gene. And this suppression of Nf2 deficiency by YAP heterozygosity is not only seen in the mouse liver, but is also observed in additional mouse tissue, such as in the eye or in the ependoma, suggesting that the YAP activation may be a general feature that underlie the overgrowth of these tumors resulting from inactivation of Nf2. In further support of this model, we found that the Nf2 deficient phenotype in the liver, shown here by the overproliferation of these oval cells, which are associated with bile ducts. And this phenotype can be greatly suppressed by overexpression of the TAD co-repressor VGL4 or by the treatment with the YAP inhibitor verteporfin, suggesting that future studies of YAP inhibitors may provide a way for the molecular intervention of the Nf2 disease. In closing, I'd like to mention that the Hippo pathway has become one of the most active areas of research in developmental biology and cancer biology in the last few years. Actually, many works from other labs have greatly expanded the role of Hippo signaling in biology. Besides, although the Hippo pathway initially identified because its important role in size control, now we know that the Hippo pathway actually plays many roles in additional biological processes. For example, the Hippo pathway has been shown to be required for the specification of the inner cell mass versus the trophic ectoderm fate in the early mouse blastocyst. This decision between the inner cell mass and the trophic ectoderm actually represents the first cell fate lineage determination in early mouse embryonic development. In the post-mitotic neuron, the Hippo pathway has also been shown to regulate events in cell differentiation. For example, the Hippo pathway has been shown to be required for the expression of specific rhodopsin genes in Drosophila photoreceptor cells. And furthermore, the Hippo pathway is required for the maintenance of dendritic branching in Drosophila sensory neurons. So therefore, the Hippo pathway is playing a much more broad role in biology, which is not just limited to size control. Recent studies from the Hippo research community have also greatly expanded the number of the repertoire of upstream regulators that regulate the Hippo pathway, both in the Drosophila system as well in the mammalian system. So I think it's a great, exciting time to work on the Hippo pathway because for most of these new upstream regulators, how they actually biochemically link to the downstream Hippo kinase cascade is yet to be determined. But the general picture here is that the Hippo kinase cascade, which we have defined, serve as a fixed signaling module which can receive inputs from multiple upstream inputs, such as cell-cell contact, cell polarity, or mechanical signal. I think it will be exciting in the next few years, and then we will be able to learn more about how the Hippo signaling pathway is regulated in these diverse biological processes from cell fate determination to organ size control and tumorigenesis. Finally, I would like to acknowledge people who contribute to this project. And all the work I described today would not have been possible without the contribution and hard work by members of my laboratory, which I've listed here. I would also like to thank several funding agencies for their generous support, which enabled us to work on the Hippo pathway using both Drosophila and mammal as a model system. I'd like to end here and take your question. Thank you very much.
Wonderful talk. I have a question regarding the pathogenesis of the Hippo pathway in human cancer. The question I have is, if the Nf2 has been shown in Hippo,
Yes.
how about the other components of the Hippo? Because it was identified in fly, they can affect size. How much do we know about their mutation, the somatic mutation in the various levels?
That's a great question. I think embarrassingly, I should say that except for Nf2, we don't have much DNA mutation for the other Hippo pathway components, except for actually epigenetic mutation changes. And we know that, for example, RAS-SF genes, which have been implicated as an upstream regulator of Hippo pathway, has been silenced in the vast majority of lung cancer, for example. So I think one of the reasons that I speculate that one of the reasons we don't find mutations for genes such as MST or LAT is because we have multiple copies of these genes. And MST, you have 2 of them, and LAT, you have 2 of them. So for Nf2, of course, we only have 1, and that's why we uncover mutations in Nf2. And I should also mention that although Nf2 was identified initially as a human disease underlying this neurofibromatosis phenotype, however, somatic mutations in Nf2 have now also been identified in other sporadic cancers outside the nervous system. So I think the role of Nf2 in growth control is beyond just neural development. Yeah.
The question is, for example, you're talking about the drug that you have been shown that has some effect. Have you tried to use, it's an FDA-approved drug, right?
Yeah.
So have you tried to use them on the Nf2 patients?
Oh, no, we haven't. No, yeah.
Other questions?
Yeah, so we believe that, okay, I should say, we believe the phosphorylation, the whole kinase cascade happens on the membrane. So the phenotype you see once it's phosphorylated, which is a retention in the cytoplasm, reflects a balance of shortening of Yorkie or YAP in 3 locations, which is plasma membrane, cytosol, and nuclear. So an analogy I like to draw here is basically you can compare YAP to beta-catenin, which lives 3 lives. It's functioning in the cytosol, it's functioning as a membrane, it's also functioning in the nucleus. That's how we think about Yorkie or YAP.
All right, I want to thank DJ and all 3 of the Marx Reward recipients for wonderful lectures this afternoon.
Thank you.
And I would remind everybody we have a reception, tea reception outside to congratulate personally each of the award winners and Dr. Marx himself.